Welcome to XenoTech's webinar, In Vitro ADME and Drug-Drug Interaction Considerations for Toxicologists. I'm pleased to have Dr. Pallavi Limaye here with us today. Pallavi received her Ph.D. in toxicology from the University of Louisiana in 2004. She joined XenoTech in 2011 as a research scientist and served as a study director for in vitro drug metabolism studies. In 2013, Pallavi joined Xenometrics, now a part of Charles River Laboratories, as a senior scientist and served as a study director for regulated non-clinical IND and NDA enabling toxicology studies. From 2018 to 2020, she worked at MRI Global as a principal scientist and oversaw non-clinical toxicology and animal health studies, as well as contributed to containment research with select chemical agents. Recently, Pallavi joined XenoTech's consulting team and provides input on drug-drug interaction study needs for sponsors. She has published several articles in various journals and has contributed many book chapters in the field of toxicology and liver pathobiology. Pallavi also serves on the program committee of the American College of Toxicology and is the vice president of Central States Regional Chapter of Society of Toxicology. It is my pleasure to present to you Dr. Pallavi Limaye.
And thanks, Michael, for the introduction. Hello, everyone. Welcome to this webinar, and I hope you all are staying safe and healthy during this COVID crisis. Today, I'm going to give a high-level overview of in vitro ADME studies and their utility in determining the drug-drug interactions for toxicologists, sort of as an introduction. This talk was originally scheduled to be given at the Society of Toxicology meeting last month, and a similar talk was given at the American College of Toxicology meeting last year. And today we are happy to bring this to you as a webinar. So while as a toxicologist, you are primarily involved in the safety assessment of a drug candidate via animal tox studies, some of you might be also overseeing the ADME side. Regardless, having the information from the ADME studies not only helps in understanding the possible drug-drug interactions, but also helps in some early decision-making for toxicology studies. So here is the outline of today's talk. First, I will discuss the rationale and objectives of ADME studies in drug development. Then I will summarize various types of ADME studies and how they provide information about potential drug-drug interaction, or DDI. After that, I will talk about the most important aspects of these studies. Those are proper study design and data interpretation, and what could be the areas of concern. Then I will talk about the timing of these studies and how this fits the overall timeline of drug development. And finally, I will conclude with some important highlights from my talk. So why run these studies? Is this just for box checking? Although a lot of sponsors are under such impression, the answer is no. These studies are pivotal in providing information on multiple important aspects of a drug. First and foremost, these studies provide a deeper understanding of a drug molecule itself, and such as its metabolism involved in its metabolism and so on. But in addition to that, if you look at the bigger picture, the main purpose of these studies is to inform the patients and the healthcare professionals about safe polypharmacy. As you know, it's a common practice to manage multiple health conditions. So most information that is generated from DDI ADME studies is used during drug label negotiations. And ultimately, the drug label guides the physician and the pharmacist to determine which drugs can be or cannot be given concomitantly, or in certain situations, adjust the dose in such a way that there are no undesired effects from the drugs. Example can be given here of warfarin. Warfarin is an anticoagulant drug with a narrow therapeutic index. Warfarin is predominantly metabolized by enzyme CYP2C9. So if there is another drug that is given along with warfarin that induces CYP2C9, it is going to increase the metabolism or clearance of warfarin and thereby decrease the efficacy, leading to likelihood of clot formation in patients. Therefore, knowing the potential drug-drug interaction is of immense importance. Now, from the pharma company's perspective, these studies helps to decide on a different candidate early on if considerable DDI liability is ascertained during these early DDI ADME studies. The results from these studies further help to determine the first-in-human dose and also the need for a clinical DDI study. And finally, most importantly, the data obtained from these ADME studies furnish the required information for drug filing as per the regulatory guidance. The three main guidance documents that are available for in vitro DDI studies are FDA guidance, which was finalized very recently in January of this year, EMA guidance that was finalized in 2013 and which is currently under revision, and the PMDA guidance for Japanese submissions, which was finalized last year. In addition to these three guidance documents, there are two other guidance documents that talk about the necessity of PK and metabolite characterization in safety studies. First one is safety testing of drug metabolites, or also known as the MIST guidance. The second revision was published just last month. And the M3 revision 2 guidance, with which all the toxicologists are familiar with, and it also talks about some of these aspects. So ADME, which stands for absorption, distribution, metabolism, and excretion, are predominantly driven by two cellular elements that can be altered. One is drug transporters that drives absorption, distribution, and excretion, and two are drug metabolizing enzymes that metabolize or clear drugs from systemic circulation. As you can see in this simplified scheme, that if one drug affects either the transporters or the drug metabolizing enzymes that handles the other drug, then it is going to result in different ADME and can result in undesired effects such as decreased effect due to decreased exposure, increased effect due to increased exposure, or cause adverse effects because of either increased exposure to the parent itself or by increased metabolism to toxic metabolites. Now, various types of in vitro studies that determine the ADME and DDI are shown here and can broadly be grouped into three subsets. The first category is drug metabolism, which drives the M and E portions of ADME and is characterized in three separate studies. The first study in this category is the assessment of comparative metabolism across various species. The second study identifies different metabolites that are formed from the parent drug and how they compare among various species. The third study, called as reaction phenotyping study, typically evaluates relative contribution of various CYPs in the metabolism. In the second category, drug is evaluated for its potential for inhibition or induction of drug metabolizing enzymes. And the third category studies the substrate or inhibition potential of the drug for drug transporters. I would mention one thing here, that these studies not necessarily need to be run in the order that is shown here. So let's start reviewing these studies first with drug metabolism studies. In the first study, a quick metabolic stability screening is performed in various species. The test system can be hepatocytes or other subcellular fractions such as microsomes or S9 from various species. This study is a very useful tool for toxicologists to determine which animal species to choose that are human relevant for IND enabling tox studies. Metabolic stability or clearance is measured in human and other typical tox species, and this list can be expanded as needed. So in this example, drug is incubated with hepatocytes from rat, monkey, dog, and human, and the drug clearance is compared. As you can see, rat and monkey are similar to humans, while dog has a faster clearance. Further, this data can be analyzed for in vitro intrinsic clearance and half-life calculations as shown here. Now, this study is also useful during lead optimization phase, where there are several compounds in the pipeline and you're trying to down select the potential candidates. The next study type is metabolite identification study. The goal of this study is to determine various metabolites formed from the parent drug in various species and figure out which species are similar to human. It also helps in identifying a human-specific metabolite. And both of these items are very important for a toxicologist because you want to select species that is similar in metabolism to humans. And you also want to know early on if there is a human-specific metabolite for which you may need to conduct independent tox studies later. So here is an example of a drug that has species-specific metabolism. In humans, coumarin is converted to 7-hydroxycoumarin, which is a non-toxic metabolite. But in rats, it is converted to epoxide, which is hepatotoxic. So the LCMS analysis identifies various metabolites as shown here in this example with ripaglinide. And on the right-hand side is a product and spectrum of one of the metabolites, that is hydroxyripaglinide. Now, one thing to be aware of when you're looking at these data is that this gives a qualitative profile because the peak height is determined by ionization efficiency and not necessarily the quantity of each metabolite. For quantitative assessment, other studies are needed that utilize either radio-level compound or reference standards for the metabolites. So the meta-ADI analysis gives a metabolic profile like this. In this example, components or metabolites 1 through 5 are formed in all species tested, whereas C6 is formed in humans and pigs, indicating that pig is a better choice of species for the tox studies. The next study in drug metabolism category is reaction phenotyping. This study typically identifies the relative contribution of various CYP enzymes in the metabolism of a drug. Drug is first incubated with recombinant CYP enzymes, and either the metabolite formation or parent loss are measured, and rate of metabolism is plotted. Now, these data are further corroborated by incubating human liver microsomes and selective CYP inhibitors, and also in a follow-up study, the non-CYP pathways are evaluated in hepatocytes. As seen in this example here, metabolism is mainly driven by CYP3A4. This drug shown here is terfenadine, which was the first non-drowsy antihistaminic drug. And in fact, it was made here in Kansas City, where we are located. When people were taking this drug alone for like seasonal allergies, it was completely safe. However, if a person also caught a bacterial infection at the same time and was given an antibiotic such as erythromycin, in some cases, it turned out to be fatal. This was because erythromycin inhibits CYP3A4 and leads to increased systemic exposure of terfenadine, leading to QT prolongation and death. Because of this, Selden or terfenadine was pulled from market due to this drug-drug interaction with CYP inhibitor drugs. And Allegra or fexofenadine, which is a metabolite of terfenadine, turned out to be a safer alternative, and it is still on the market. So overall, for avoiding potential DDI, usually drugs that are metabolized by multiple pathways are preferred. Because if one of the enzymes is inhibited, there are other pathways that can metabolize the drug. Here I would like to introduce you to two terms. Those are commonly used in DDI, and they are victim drug and a perpetrator drug. In this example that we just saw, terfenadine is a victim drug and erythromycin is a perpetrator drug. So this study type, that is reaction phenotyping study, determines the victim potential of a drug. Now moving on to the drug metabolizing enzymes, there are two studies that are inhibition and induction studies that are conducted to determine the perpetrator potential of a drug. Historically, there are more victim drugs that have been withdrawn from the market than the perpetrators. For CYP inhibition study, human liver microsomes and a marker substrate of a particular CYP are incubated with or without pre-incubation with the drug. And further, IC50 values are calculated to determine the clinically relevant CYP inhibition. So here is a list of preferred activity assays for the corresponding CYP enzymes. And here are two examples of CYP3A4 inhibitors. Ketoconazole, shown on the left, is an antifungal drug that is either pre-incubated with human liver microsomes prior to starting the reaction with marker substrate or was added directly with the marker substrate. And CYP3A4 activity is plotted on y-axis, and the concentrations of ketoconazole are plotted on x-axis. As you can see, the two curves pretty much are superimposable with no appreciable difference in the IC50 values with or without pre-incubation. And this indicates that ketoconazole is a direct inhibitor of CYP3A4. The mibefridil, on the other hand, has a significant leftward shift in the plot, indicating that this is a time-dependent inhibitor. Now, why is this important? It is important that if it is a direct inhibitor or a time-dependent inhibition, because time-dependent inhibition has a greater DDI risk. Direct inhibition persists only until the parent is around. However, time-dependent inhibition lasts much longer. In some cases, in fact, it requires synthesis of a new enzyme before that CYP becomes functional again. This was the case with mibefridil, and therefore it was withdrawn from the market due to its potent and persistent inhibitory effect. Mibefridil also represents an example of a perpetrator drug being withdrawn from the market due to drug-drug interaction. For CYP induction studies, human hepatocyte cultures are incubated with the drug, and then mRNA is measured. The regulatory guidance requires cultures from a minimum of 3 donors to account for any inter-individual variability. If positive results are obtained in any culture, the drug is considered as a positive inducer. Further, Emax and EC50 values are calculated for each culture to determine the potential for induction. Now, for both inhibition and induction studies, regulatory guidance documents have suggested some methods to extrapolate in vitro to in vivo determinations, such as the basic or mechanistic models, and the data generated in our inhibition or induction studies allow sponsors to conduct such analysis. Now, moving on to the drug transporters, the studies are conducted to evaluate the substrate potential, which is a victim potential, or studies are also conducted to evaluate the inhibition potential or a perpetrator potential. So as shown here in these schematic representations, there are several uptake and efflux transporters that transport drugs either inside of the cells or the parent or metabolites outside of the cells. The transporters those are important from the drug development standpoint are the ones shown in boxes. So for these studies, either polarized cell lines that express the specific transporter or vesicles are utilized. For substrate potential, transport of the drug is evaluated with and without the known inhibitor on board. For inhibitor potential, transport of a marker substrate is evaluated in the presence or absence of the drug. So as shown here in this example, apparent permeability and efflux ratios are plotted on y-axis, and the inhibitor concentration is plotted on x-axis. As the inhibitor concentration is increased, the basal to apical transport is reduced, and so is the efflux ratio. So depending on the study type, this determines the substrate or the inhibitor potential of a drug with a similar principle for these two studies. So now that we have reviewed different types of in vitro ADME studies, let's consider some examples of areas of concern. So the first example here is of CYP inhibition study design. If you do not design the study properly, you can end up with false negative results. So as shown in this example, clinically relevant time-dependent CYP2C19 inhibition by omeprazole or Prilosec was missed for many years due to the use of high protein concentration and longer substrate incubation time. As seen on the left, when the study was conducted with 1 milligram protein and 30-minute incubation, similar IC50 values were observed, and it was missed that omeprazole is a moderate time-dependent inhibitor. But when lower amount of protein and shorter substrate incubation was utilized, a leftward shift in the curve was observed compared to no pre-incubation and pre-incubation without an ADPH, revealing time-dependent inhibition by omeprazole. Now, why is this important? As we saw before, this info is important for the drug label. Time-dependent inhibition has a higher risk of DDI than a direct inhibitor, and therefore designing the studies properly is very important. The next example is of a CYP induction study. Testosterone hydroxylation is plotted on y-axis as the marker of CYP3A4 activity, and various cultures are ranked based on their testosterone hydroxylation activity on y-axis. Now, after rifampin incubation, which is a known inducer of CYP3A4, the cultures are plotted again for testosterone hydroxylation activity. And as you can see here, the cultures in the box, gray box, have very high induction as shown in the third figure here. However, their basal activity itself was very poor, leading to such supraphysiologic increases of several hundred folds. Now, these data indicate that these false results are because the cells are not fully confluent, or maybe there are gaps in the culture. Such data should be the ones that we should be suspicious of. Good cultures have high control CYP activity, and this lowers the magnitude of induction to something that is physiologically possible up to like 20 folds, not hundreds of folds. So here is an example that shows that when it comes to induction, more is not always better. In fact, when the cultures are poor, there is a possibility that induction potential may be even missed for your drug. So this example underlines the importance of proper interpretation of your data. The third example here is of reaction phenotyping study. Choosing the right test system based on the structure is very important. So as mentioned before, a lot of sponsors are under the impression that these studies are only for box checking and insist that they only want to do study the CYP panel. But look at this example here. Ezetimibe can of course be metabolized by CYP3A4 when incubated with human liver microsomes and NADPH. But in reality, clinically, this is very misleading. Because if you look at the structure, there is this phenolic hydroxyl that can be readily glucuronidated. And even before, CYP metabolism can occur. And there is very little parent compound left for CYP3A4 metabolism. Therefore, in this case, studying the role of UGT enzymes is more applicable than the CYP panel. The point to remember here is that CYP is not the only enzyme system, and depending on the structure of the compound, other enzymes and thereby test systems also need to be considered while designing the studies and interpreting your data. Now that we have reviewed types of in vitro studies, how to design the studies, now let's look at the timing of these studies against the whole timeline of drug development. So this is a sort of a busy slide, but you can see that there is a lot of subjectivity when it comes to the timing of these studies. Usually it is advised to get as much data as possible early on in the development program. However, in practical world, the priority depends on how much de-risking is required for each individual molecule and the sponsor needs. So as shown here in this table, for small molecules, typical small molecules, it is preferred to look at the metabolism studies during lead optimization, CYP induction and inhibition, transporter inhibition, and substrate potential of some transporters should be done prior to IND filing, and reaction phenotyping and substrate potential for the remaining transporters can be deferred until the phase 1 to NDA timeframe. For small molecules with orphan or breakthrough status and peptides and oligos and other biologics, Some of these studies can be deferred until later and can be run during the phase 1 to NDA timeframe. So the most important point to be noted is that you want to collect as much DDI information on your drug as possible so that you are not unnecessarily excluding patients in clinical trials who are on different drugs, as this may lead to delay in the approval as stated in the FDA guidance. So in a nutshell, the timing really depends on your own risk tolerance and your de-risking strategy. So to conclude, why are the in vitro studies important? It is because other than revealing the DDI potential, they give you an insight into future performance in vivo studies, such as right test species selection for tox studies or first in human dose selection. They satisfy the regulatory interests and prevent unnecessary delays in approval. Now, timing depends on the drug class and your own de-risking strategy. As we saw from the examples of areas of concern, conduct and interpretation can be deceptively simple, but they both can benefit from expert design and understanding. And finally, I would like to remind sponsors to provide as much information as you can about your drug to the scientists, those will be conducting these studies. That way, they will be able to guide you better when they have all the necessary information, such as the structure of the compound. So with that, I would like to end my talk, and I will be happy to address any questions and receive any comments. I would also like to mention that in addition to me, I'm fortunate to have Dr. Brian Ogilvie, the Vice President of Scientific Consulting here at XenoTech, to answer any of your questions with his more than 20 plus years of experience in this field. So thank you for listening to my talk, and I'll be happy to take any questions.
Thank you, Pallavi. And while we wait for questions, I wanted to mention that information about our services and test systems, published papers, posters, other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Pallavi's presentation or any of Sekisui XenoTech's contract research services or products, you can also ask them in the post-webinar survey or contact us through our website.
Okay, hi, this is Brian Ogilvie. Pallavi, thanks for the brilliant presentation today. And I'm trying to sort through a couple of these questions here real quick. So there's one question that came in saying, for CYP inhibition and induction conducted prior to IND, we typically don't have information on clinical drug concentrations. So what concentrations do you recommend for these studies? And Pallavi and I were actually on a call yesterday discussing this very topic. It's very common. And so what happens is we will take information that we already know about the compound. It may be simply what the projected dose is, if you have that. And if not, then in many cases, because of drugs that are developed today have low solubility, solubility limits may be the answer. And also, you need to consider cytotoxicity. So some cases we will actually perform studies preliminarily to determine A, what is the solubility in the test system, and then B, what is the cytotoxicity limit that we can reach. And those may be simply the only concentrations that we can reach as a highest limit.
Use the concentration that is maximally feasible. So depending on the aqueous solubility of the compound, we can use the maximum concentration possible, just like what we do for maximum tolerated dose, where we use maximum feasible dose.
Okay, and then there's another question here about if we have a compound that's very slowly metabolized, do we have any recommendations on a system to use? So this is something that you have to define, first of all, what is slowly metabolized. And I think a lot of times we have clients that will come to us with a compound that they may have done some in-house data showing that it is so low turnover compound. And when we go ahead into hepatocytes, we find a reasonable rate of turnover. However, there are certain ones that are, of course, very low turnover compounds. And in those cases, we've found that pooled plated hepatocytes are a good system for this. and we can get incubation times up to 48, even 72 hours, and have determined in vitro clearance values that correlate well with the clinical values. What are the general guidelines for appropriate DDI testing for testing pharmacologically active metabolites? So the FDA guidance that was just finalized in January essentially says that if you have, I think they put it in terms of significant pharmacological activity, that you need to examine those metabolites also as, as if it were the test article itself. And so if you know you have a metabolite with significant pharmacological activity, you're just going to have it synthesized and run it through most of the same battery of tests. One exception to this might be in hepatocytes for induction studies. So if you can show that the metabolite is formed in the hepatocytes, then you're essentially testing both the metabolite and the parents at the same time in the induction study. What about repurposing drugs, and are there special considerations? So there's another pathway, as I'm sure many of you are aware, for the typical pathway for new drug applications is a 505(b)(1) filing, and that's the formal, application that would be complete. The 505(b)(2) pathway, however, would be used typically for repurposing, and that's a case where you can take much of the legacy data that exists. And I've worked on a number of 505(b)(2) applications where what we do is we take a look at the legacy data and see if there are any gaps that need to be filled. And sometimes there are, and you just work on a risk-based approach to fill in the gaps, and then those can go into the 505(b)(2) pathway application.
Yeah. And I think the next question, if we see a borderline induction, is it necessary to conduct clinical DDI?
Yes, although I would add to that on the borderline induction. So, say we got predict a value of 0.79, something like that. From what I've seen, the agencies will take a strict interpretation, even if it's only in a single culture, that you would need to do some further testing. And that could be in the form of not only a clinical DDI, but as I mentioned, the full PBPK modeling that can occur, which will often allow you to mitigate the potential for that positive effect and maybe avoid a clinical drug-drug interaction study. Okay, here's a question on if time-dependent inhibition study is required for any drug metabolizing enzyme inhibition study. So CYPs show a greater tendency to be inhibited, and how about other drug metabolizing enzymes such as UGT and SALT? So I mean, my focus for most of my career has really been on metabolism or time-dependent inhibition of P450s. And the reason that occurs is largely because you have a metabolite that is not necessarily released from the active site, and so it irreversibly inactivates the enzyme. There are other mechanisms by which that can occur. With other enzymes, I think it has been described on occasion, but it's extremely rare as far as I know. I will also say there are some, and I published on this in 2006, cases where UGTs may actually be involved in metabolism-dependent inhibition of CYP2C8 in particular. So for instance, gemfibrozil is metabolized by one of the UGTs, and its glucuronide metabolite then and inactivates CYP2C8. Okay, here's one on, is it always assumed that as a class such as statins that they will act the same if there's demonstrated drug-drug interaction with one? And I would say absolutely not, and that comes down to structure. You can make some guesses. So for instance, with statins, most of them tend to be substrates and therefore competitive inhibitors of certain transporters such as OATPs, but not always. And so if you look also at statins, one famous example was cerevistatin, which was removed from the market because when it was given with gemfibrozil, as I mentioned, the glucuronide of that inactivates CYP2C8. So cerevistatin is metabolized by CYP2C8, whereas several other statins are metabolized by CYP3A4 and glucuronidation as their major pathways.
Okay, it sounds like this is going to wrap up today's session. Thanks to all of you for taking time out of your day to join us, and thank you to Pallavi for that great presentation. Have a great rest of your day.